is DNA like a computer? Well, we know that computers require intelligent design, don't they? And there are many aspects where DNA seems to function like a computer, and not just based upon the codons and the codes that send DNA. There's a lot more to DNA, just more than just reading those codes. And Stephen Meyer points this out in his book, Signature in the Cell. There's multiple messages, for example, that can be stored in the same sequence of bases. And sometimes these messages overlap. So you'll have same message that overlaps with each other. There's no really good evolutionary explanation for that. How would evolution explain that? And yet we know that's how DNA works. There's splicing, there's editing, there's reading processes that can produce more than one protein from the same RNA message. And that's profound, really. And that's one reason, or a reason why mankind may have 23 pairs of chromosomes, and yet some organisms have quite a few more chromosomes. And it's because of the splicing, it's the editing, it's the reading processes, and the fact that one protein can come from the same RNA message. And so very computer-like in the way DNA is structured, again showing intelligent design. W.Y. Chung, a bioinformatician, hard to say that word, at the Center of Comparative Genomics and Bioinformatics at Penn State University, has stated, he has noted the existence of dual coding and overlapping, what we've just been talking about, and protein coding reading frames is virtually impossible by chance. It just wouldn't happen by chance and by naturalistic processes. And yet that's what we see when we study DNA. We see this splicing, we see this editing, and it's just not likely chance would explain it. Genetic messages are encoded within genetic messages, and that's been likened to an encryption device like we had during World War II, where we were able to decipher the Germans' messages and probably help win the war. Well, there's things like that in DNA itself. It's encrypted. And these messages that we're seeing in the DNA molecule. And then there's, so we can see that it's very computer-like. Now this is a part that you don't hear, that we're going to talk about the orphan genes, okay? In reading Dr. Collins' book, I never heard him mention one thing about orphan genes, because orphan genes, just think of it this way, it's sort of the exact opposite when you start looking at similarity of chromosomes. The evolutionists want to look at how similar animals' chromosomes look to each other, how we have similar chromosomes to a mouse, similar chromosomes to a chimpanzee, and so forth. What they don't want to look at is orphan genes. Well, what is an orphan gene? The word comes from open reading frames of unknown origin. That's an evolutionary term, obviously, unknown origin. We know where their origin is. But for them, it doesn't make sense, because what orphan genes are hundreds of thousands of genes that do not exhibit any significant similarity in sequences to any other known genes. And these genes have no homologue and cannot be explained by evolution. So what am I saying here? I'm saying you have genes that other animals don't have, and they have genes that you don't have. But according to evolutionary theory, we should be sharing a lot of those genes. Well, they can't explain orphan genes. So there's literally hundreds of thousands. So you can't have it both ways. You can't say, well, look at the similarity that we have. Look how similar our genes are, and ignore orphan genes, because orphan genes show how dissimilar we are. Because there's genes found in some organisms that aren't found in any other species. And for evolution to occur, that doesn't make sense, does it? We should be sharing these genes. And yet the orphan genes are a fact. Now some have argued, I don't have this on the slide, but some have argued, well, maybe we just haven't found enough of these orphan genes and we haven't discovered their similarities yet. But as more and more orphan genes are found, we understand how dissimilar they are. And that's a bad argument. It doesn't work. I want to look at NeoDarwinism and math just a little bit. And I know we've looked at a lot of big figures. Last time we met 2 weeks ago, there was a lot of big figures I threw at you. Well, we're not quite through yet. I want to look at just a few more big figures. But I wanted to talk about one more topic before we do that. And we talked about, remember the molecular clock, and you remember the supposed evolutionary tree that was generated by the molecular clock. And almost all of these are computer-generated programs, right? And we looked at Dawkins' methinks it's like a weasel, how he was able to get that computer to come up with that all on its own, trying to show the similarity, how life could have begun. But there's a problem with all these, what Stephen Meyer calls genetic algorithms. And here's the problem, when we talked about this last time a little bit, and that is they lack realism because by providing the program with a target sequence. So you have the target you're reaching for. That target's already been determined. Evolution doesn't have a target, does it? I mean, evolutionists wouldn't say there's a target. Where's the end point? I mean, they would say we're still evolving, wouldn't they? You know, where are we going to be, you know, a million years from now? According to the evolutionists, there's no target. But when they come up with these algorithms, these genetic algorithms, they always give it a target. Well, that's unrealistic. That's not how evolution works. Furthermore, by programming the computer to select for proximity to future function rather than actual function. So they program it to try to go into the future, to go towards that target. And that's not real. And by selecting for changes that fail to model biologically realistic increments of functional change, increments that reflect the extreme rarity of functional sequences of nucleotide bases or amino acids in the relevant sequence space. So all those genetic algorithms, and when you read these books, and if you're a student and you read these programs and show how evolution had to have occurred because of these computer programs, this is the problem you have. It's just not a fair thing. Now we're going to go into NeoDarwinism and math. And I want to look at a few equations that are just difficult to explain. First of all, a 300 amino acid length protein can represent just one possible sequence. Now remember, 300 amino acids represents about 1,000 base pairs, because you need 3 base pairs to equal 1 amino acid, and those amino acids have to be linked together to form the protein. And they have to be linked together in a very specific order. It's sort of like, again, flipping the coin and getting heads one time and tails the next time, and then heads and heads and tails, and coming up with that sequence. So that's what this number is representing. In order to get exactly those 1,000 base pairs, or looking at the other way, 300 codons in the exact same order, the probability of that sequence is 20 to the 300th power, or 10 to the 390th power. That's a very, very unrealistic number, as there's only like 10 to the 69th power of atoms in the Milky Way, our galaxy. That's how unrealistic that number is, and that's just for one protein. And think of all the proteins our bodies have. So that's just, I think, causes one to pause and see what how realistic it would be for those amino acids to line up in the right order to produce that protein. And then Michael Behe, who we're going to look at in some detail later on in this course, he's the one that came up with the irreducible complexity idea, and him and David Snook calculated that mutations in selection could possibly generate 2 coordinated mutations in a mere 1 million generations, and it would take a population of 1 trillion or more multicellular organisms to do this, or a number that is completely impractical and exceeds the size of breeding populations. We don't talk about trillions of populations, do we? It just is totally unrealistic. Conversely, they found mutations in selection could generate 2 coordinated mutations in a population of 1 million organisms, but only if that mechanism had 10 billion generations at its disposal. And if you consider the fact that 10 billion generations represents 1 year, how old is the Earth, supposedly, according to the evolutionists? 5 billion years? And that's for one change. That's for one change. So you couldn't get 1 mutation change to produce a different organism in 10 billion years. Or you could get 1, but you couldn't get enough to make an organism. since 10 billion generations computes to 10 billion years, twice the supposed age of the Earth. This represents an unreasonable length of time to wait for the emergence of a single gene, let alone any other significant changes that could occur evolutionary. So to get that one gene change would supposedly take 10 billion years. Now, we don't believe that the Earth's 5 billion years old. I mean, I don't. So, but as we started at the beginning of the class, even if you hold to that, there's not enough time. Give it 30 billion years. Give it 100 billion years. There's not enough time to do these changes. If mutations could even cause a change in an organism, which we've already seen could not change one organism from another organism. Now this irritated a couple of people at, I forgot what college they were at, Rick Durrett and Dina Schmidt. They wrote kind of a rebuttal and did some calculations of their own after reading Michael Behe's book. The Edge of Evolution is where this data came from. He's also written the book Darwin's Black Box, and some of y'all I'm sure have read that. Well, they looked at these and go, well, those calculations are they're absurd. And so they wrote a paper waiting for 2 mutations with applications to regulatory sequence evolution and the limits of Darwinian evolution. And the calculations suggested it would not take several 100 million years, but, quote, only 216 million years to generate and fix 2 coordinated mutations in the hominid line. The problem with that is that's a long time. That's a lot longer when you start looking at the number of mutations that supposedly occurred, the millions of mutations that had to occur to get from chimps to humans. You're not going to have enough time. They proved the point. They were trying to rebut it, but they proved. Now, their numbers aren't as big as Michael Behe's, but it doesn't matter. They proved they proved Michael Behe's point and didn't realize they did it. This is more than 30 times the amount of time available to produce humans and chimps when you look at all the supposed mutations that occurred to get from chimps to humans. So calculations performed by both critics and defenders of neo-Darwinian evolution actually reinforce and basically come to the same conclusion. Now, we're going to look at cytochrome C. Now, I don't want to get too technical, but I thought this was fascinating, so I'm going to spend just a little bit of time on it. Virtually all animals have this protein. It's called this molecule, cytochrome C molecule. It has 100 amino acids, therefore it is a protein, and it forms a 3D configuration. And the amino acid sequence varies from organism to organism, but it's found in bacteria, it's found in fungi, it's found in higher plants, and it's found in animals, including you and me. The cytochrome C molecule. And evolutionists will say, well, see, here again, they want to ignore the orphan genes, but they want to look at this and say, look, we all have this. All animals have this cytochrome. This shows that we came from a common source, where it came from evolution. You can even go to Dayhoff's Atlas of Protein Sequence and Structure, and it categorizes 1,089 entries into a matrix form for cytochrome C for all the various animals that are out there, and plants, and living tissue that has this cytochrome C. And this is current. You could go look that up. As an organism increases in distance from one another, their amino acid sequence varies more and more. So they have the same cytochrome C, but the sequencing is completely different from organism to organism. Does that make sense? The sequencing is different. What's interesting, though, is that there are no transitional or intermediate classes from one organism to the other when looking at cytochrome C. You would expect it, in other words, to be a gradual change, but there's not. They are absent, as Michael Denton points out. They are absent from the matrix. They're not there. There's no transitional intermediate classes. All organisms, from humans to yeast, exhibit sequence diverging by 64 to 67%. You're just as close to a fungi as you are a reptile, based upon cytochrome C. That divergence. I know that's a little technical, but it's an amazing thing, and it's not explained evolutionary at all. And as Michael Denton points out, this is almost mathematical precision. The isolation of fundamental classes of organisms at the molecular level, there is not a trace at the molecular level of the transitional evolutionary series, from cyclostome to fish to amphibian to reptile to mammal. Incredibly, man is as close to the lamprey as are the fish in this divergence pattern. It makes no sense evolutionary, and yet the evolutionists want to use it as one of their defenses for evolution. I thought that was fascinating. As Denton points out, thus there is incredible orderliness in all divisions biochemically, a phenomenon that fits perfectly where there's an intelligent designer. All right, who watched the debate between Bill Nye and Ken Ham? Surely more than just a few of you did. Okay, well, you should have watched that debate. You can still go watch it if you want to. They can still watch it. I thought it was a great debate, but what came out in that debate that Bill Nye kind of drove home to Ken Ham? He kept saying that evolution makes predictions. Do you remember that, those of you that saw the debate? Evolution makes predictions. Intelligent design, he didn't use intelligent design, but creationism does not make any predictions. Well, Ken Ham said, yes, we do, and he gave a few examples, but I thought personally that he dropped the ball a little bit in that debate by not coming up with more predictions that, and of course it's a debate, thinking off the top of your head and everything else had to be tough to do, and you might not have known where that question was coming from. The point is that to criticize creationism because we don't make predictions, or it does not make predictions, or intelligent design because it doesn't make predictions is just not true. There are predictions that you can make based upon intelligent design, based upon creationism. And so, again, I think I thought it could be handled a little bit better, but so I'm going to try to kind of rebut Bill Nye a little bit today to show that there are predictions that can be made, and some of the predictions have already been shown to have been true. And these predictions were made before they occurred. And like I say, the truth is there are quite a few predictions that intelligent design can make, many that would not be predicted by naturalism. What are they? And there's about 12 or so in Stephen Meyer's book, Signature in the Cell, and there's more than that, but he just put those 12 in there. And one of the predictions is that the fossil record will continue to show, and does show, a top-down phenomenon and not a bottom-up phenomenon, which is in fact has been verified. In other words, when you look at the fossil records, you see, and he's written another book called Darwin's Doubt, which deals with the Cambrian explosion. Have you heard that term before? And what happened in the Cambrian age, if you buy into these ages that the geologists have come up with, a lot of multicellular organisms show up all of a sudden at the bottom of what we consider the geologic column without any transitional forms to get to that point. You understand what I'm saying? They're just there all of a sudden. Now, when I was going through school, I had the idea that Cambrian, everything showed up. Well, no, that's not true. But there are many, and most of these animals are extinct, if not all of them. But they're multicellular organisms. They are diverse organisms, many diversities, and they all show up all of a sudden in the Cambrian age. That would not be predicted from an evolutionary point of view. And that's just another way, that phrase I've put there, that's just another way of saying that. That's not what you would expect evolutionary. You would expect to go from one cell to a few cells to maybe finally a 100-cell animal, and then so on. It would take millions of years for that to occur. That's not what you see in the fossil evidence. And evolutionists they really don't have a good explanation for it. Their best explanation for it is, well, we just haven't found those fossils yet. That's their best one. Or many soft-shelled or soft-bodied animals would not fossilize, but that's not true either. We've got fossils of bacteria, so that doesn't make sense either. But that's their answer. So that's one prediction that creationism would make. Another prediction is, and we've already seen this, creationists predicted junk DNA would be found to have functionality. Now, do you remember, and I know this was a few weeks ago, but you remember the quote that I gave you of William Dembski? William Dembski had predicted that you would find that DNA had functionality before the human genome came out. Before that. So the human genome comes out, or the genome of other animals too, And evolutionists look at all this junk DNA and they go, look at there. See, this was predicted by evolution. But they spoke too soon, because we're now finding that that supposed junk DNA really does have function. And we listed all that in our previous lesson, so I don't want to go over that again. But this was a prediction that creationists made, and it's turning out to be true. Intelligent design and creationism also predict that organisms have limits to change. And that's just proven observationally. When you're doing breeding of animals, domestic animals, for example, you know there's a limit you can get to. You can't get beyond that limit. And that's a prediction that creationism would make, and it's observationally true. You cannot. You can get a dog so big and so small, and there's a wide variation, but it's still that dog, isn't it? There's a limit to what they can do. Jonathan Wells, this I thought was really fascinating personally, because this is something that truly has practicality to our lives. Jonathan Wells, reasoning from a design point of view, postulated that the centrosome, a cellular organelle, might be the culprit in the cause of cancer in some cases. Scientists have primarily honed in on chromosomal damage as the cause of cancer, right? Or you understand that? And so they really haven't paid too much attention to what other causes there could be. It's damage to the chromosome. In other words, a mutation in the chromosome that causes cancer. Well, again, Jonathan Wells, reasoning from a design point of view, he would not have come up with this had it not been from a design perspective, because you wouldn't think of this evolutionary. But this cellular organelle called the centrosome, and I can't tell you exactly, it's been a while since I've studied my biology, what centrosome does, but it's an organelle in the cells of our body. But it's possible that this could be the culprit of causing at least some forms of cancer. Well, it's turning out to be true. And now we have scientists that are going back, and the people that do research in cancer and so forth, that are starting to look at that. Not all cancers, not saying that, but at least in some, the centrosome may be the culprit. Then you have our friend again, Michael Behe, reasoning from the in irreducible complexity point of view, predicts more and more mini machines within the cell will be discovered that carry on more functions than we currently realize, and they're starting to find some of those mini machines in the cells. These mini machines, such as the flagella mentioned in Behe's book, Darwin's Black Box, demonstrate incredible design. We will be looking at that. We'll be looking at the flagella and the design of the flagella, and how could you possibly ever get a flagella from an evolutionary point of view? You could not. So to criticize creationists in the intelligent design community for not being able to make scientific predictions based on their concepts is patently wrong. And there's many more we could have gone over, but for sake of time, we're not doing that. All right, we're going to finish up in looking at apes and men, because I know people have probably asked you this before. Maybe they haven't, but they have me. And that is, well, how do creationists explain the similarities between apes and man? That's a good question, don't you think? You know, from an evolutionary point of view, I mean, hey, you've seen those iconic figures that are in all the textbooks that start off with a hunched over animal that looks like an ape or chimp, and progressive, and we're going to look at that too, by the way, in some detail in future studies. But you've seen those progressions, and then you get closer and closer to man. You get to Neanderthal, then Cro-Magnon, and so forth, and voila, you've got modern man walking around now. And so there are similarities. And so how do we explain that from a creationist standpoint? Well, quick answer for me is this. Does it not make sense that if an organism looks a lot like another organism, at least in some cases, that there's going to be a certain amount of DNA that's going to be similar to? What would be so profound about that? I mean, apes have two eyes. Most animals have two eyes. You don't see too many one-eyed horses or other animals, you know. But, I mean, they have two eyes, you know, four limbs, so on and so forth. Would it not make at least some sense that if they're going to look a little bit alike, that the DNA would probably be similar too? I mean, it just makes sense to me from that point of view. So I don't think it's all that you know, that big a deal. That there's similarities is really not the question. There are similarities between men and chimps, and in man's and chimps' DNA. That's not the question. The question is, what does the similarity mean? We go back to the homologue again, the homology. Remember that? And that Darwin's only really defense for evolution was the homologue point of view. But that doesn't prove anything. But are there similarities, and how significant are there similarities? And we're going to spend a little time talking about that. And is evolution an adequate explanation for the similarities or for the differences? So before the completion of the Human Genome Project, starting in 1965, Mary Claire King at the University of California, San Francisco, and Alan Wilson, University of California at Berkeley, argued that there was only a 1% difference between chimps and man's DNA. Now this surprised even them, because morphologically there's more than 1% difference. You know, there's more difference just morphologically, looking at the animal. But the DNA, and I'm not sure how they came up with that, but it turned out to be fairly close. In 2005, Pascal Gounod reported researchers were finding out that the 1% distinction might not be all that accurate. Chunks of missing DNA, extra genes, and altered connections confound even any qualification of humanness versus chimpness, changing this number from 1 to 3%. So when the genome came out, it was originally thought that they had proven these other two scientists correct. But turns out there was a little bit more difference. But 3% is still pretty close, wouldn't you say? Kind of. Sounds pretty close at least. 97 versus 1. And this number changes a little bit depending on who you read. You're not going to get the same number every time you read that, but you're always going to get somewhere between 1.3 to 3.4% when you read the science here. So within the differences of the numbers of copies of DNA, this number becomes as much as 6.4% when you look at differences in the number of copies. And the greatest difference, as you might suspect, between chimps and humans is the code that's coded for brain function. Excuse me. That's where we see the biggest differences. So let's look at a little bit of the differences in the DNA between chimps and humans. The genome for the Y chromosome in the chimpanzee was not completed until 2010. In your handout, by the way, I got the dates wrong. On one of the human genome was completed in 2003. I think I have 2013 there. The chimp genome was completed in 2005, but the Y chromosome for the chimpanzee was not completed until 2010. You know what the Y chromosome is? That's the chromosome in men versus women. Well, they looked at the Y chromosome in humans. There were 78 genes in the Y chromosome, and the chimp only had 37. That's a 58% difference. There is absolutely no explanation for a 58% difference between the Y chromosome between the chimps and humans. So that's a pretty big difference. Now, let's look at the number and how big a difference is 1%. We're talking about 1.23% here, and most people would agree it's at least 3% difference now. It's not 1%. But even if it was, 1.23% difference in genes represents 35 million differences. It's a big difference and would take a lot of time evolutionary to get there. To give you a perspective of this, if you were to have an 8.5 by 11 page, it can hold 4,000 letters, and it would take 10,000 pages to equal 40 million letters, or in other words, the base sequences. That's a lot of pages, and that's on 1.3% difference. So if you triple that, then you would see 30. In other words, 1% may not seem like a lot, but 1% is a lot. And from an evolutionary standpoint, would take a long time to get there. So is this a lot? I just would say this, it's in the eyes of the beholder. But from an evolutionary standpoint, it's a whole lot. Further differences, I'm going to go through this pretty quick because I want to try to finish this lesson up. Further differences involve telomeres, repeating sequences at the end of sequences. Chimps have 23,000, humans have 10,000. Humans have 689 genes, chimps lack, and chimps have 86 genes that humans lack. Quite a bit of difference. And if you realize how long it takes, remember earlier tonight we talked how long it would take to get from just to produce one new gene. It's a long time. Small differences in gene sequences can mean a large difference. For example, the FOXP2 protein is said to be involved in language. The reason we can talk and chimps can't talk is supposedly because of the differences between the FOXP2 protein. What's the difference? We're identical to 99.7%, and yet I haven't seen a chimp talking yet. Also, humans have 23 pairs of chromosomes, chimps have 24. Now, the evolutionists explain this by saying, well, in humans, 2 of the chromosomes fused, so you have 1 less. They don't have a good reason for why they would have fused. So again, they say that they were fused. And there's no good evolutionary explanation. Finally, evolutionists point to the similarity in junk DNA shared by chimps and humans. But once again, there really isn't any junk DNA. Haldane's dilemma. Now, Haldane is not a creationist, or was not. He's not alive now, but he was not a creationist. But he understood there was a dilemma when he looked at the difference between chimps and humans and how much time it would take evolutionary from a mutational standpoint, in other words, a NeoDarwin standpoint. And per evolutionist, 40 million total separate mutation events have separated humans and chimps. Haldane calculated the average mutation rate of 10 to the 6th power, which is consistent with evolutionists. Given evolutionary timescales, this implies, I don't know what that number is, it's 150 followed by 9 zeros. Anybody know what that is? 150 billion. Okay, there's 150 billion forerunners to modern man. Does anybody have a problem with that? Where are the fossils? That's a lot of fossils that we haven't found yet. It's not just fossils, but if you look at it from population study standpoint, there's not enough time to have produced that many forerunners. And so the obvious question is the fossils, and they should be innumerable. Population studies would also make that number utterly impossible. So what's known as Haldane's dilemma. And again, he was an evolutionist. We'll be looking at him next week a little bit as well. There cannot be, and there are not, thousands of millions of pre-humans. They're just not there. How can evolutionists even explain this? Well, go back to, remember, abductive reasoning. We looked at it in one of our very first classes. If P, then Q, Q, therefore P. That's the evolutionist logic. It's not our logic, but it's theirs. In other words, if humans and chimps share a common ancestor, there will be evidence of chromosome similarity. There is chromosome similarity. Therefore, humans and chimps share a common ancestor. See a problem with that logic? I do. It's not really logical. It's making a conclusion you really can't make. So are there similarities between humans and chimps' DNA? Yes, there certainly are, but there are also, as we've shown, significant differences. Even from an evolutionary point of view, there simply isn't enough time for mutations to have produced humans from chimps, even if mutations were capable of doing so, which we have clearly seen they are not. So even if you could do it by mutations, there's not enough time. Overwhelmingly, design and common designer is the explanation for the similarities between chimps and humans. So summarizing, we've looked at DNA, genetics in the genome. They offer no more proof for evolution than homology did in the past or the fossil record. It's okay, there's similarities, same as the argument for homology. I would say designer has a better explanation. And it's very presumptuous. This is key. This is key to me. It's very presumptuous to say evolution has occurred when it's impossible to determine how even a single functioning protein could have ever occurred naturally. You can't even prove one protein occurred naturally. And you're going to tell me we got here naturally without any input from an intelligent designer? It's very presumptuous. NeoDarwinism fails. This is from Stephen Meyer. I'll go through this quickly. Because it has no means of efficiently searching combinatorial sequence space for functional genes and proteins, and consequently it requires unrealistically long waiting time to generate even a single new gene or protein. It has also been shown that the mechanism cannot produce new body plans because early acting mutations, the only kind capable of generating large-scale changes, are also invariably deleterious. And epigenetic information necessary to build the body plan, there's no explanation for that, and we looked at that. DNA, mRNA, proteins are identical and have no living system that can be thought of as primitive. In other words, you look at the DNA in a bacteria, you look at the DNA in a human, there's a lot of similarity there. And they're not primitive. One didn't evolve from one to the other. And they carry on very, very similar functions. That's not an argument for evolution. That's an argument for design. Bacteria cell, look at the bacteria cell. You can do that sometime and magnify it 20 times, and it's unbelievable. It looks like a giant ship large enough to cover the city the size of New York with many functions. It looks like a major refinery. And that all occurred naturally. Michael didn't. It is the sheer universality of perfection. The fact that everywhere you look, to every depth we look, we find an elegance and an ingenuity of an absolutely transcending quality which so mitigates against the idea of chance. I could not have said it better. We've got one more slide, I think. The case for design restores Western thought to the possibility that human life in particular may have a purpose or significance beyond temporary material utility. No joke. And I know what that purpose is, don't you? It's in the book. And that was the best part of that debate in my mind. Every time Mr. Nye could say, well, we don't have an explanation for it. Remember what Ken Hemp would say? He would say, I do. It's in the book. And it's in the book. Thank you. Next week, we'll be moving on to another topic.